Good morning, everybody. On behalf of the GI Cancer Center at the Mass General Cancer Center, I want to welcome you to our exciting webinar this morning on innovations in esophagogastric cancer. We're very excited to have you joining us. My name is Dr. Ryan Corcoran. I am the director of the GI Cancer Center program here at Mass General and also the scientific director of the Tremere Center for Targeted Therapies, and I'll be your host for this exciting event. So before we get started, I just wanted to review a few quick housekeeping items. So there will be no microphone or video capabilities needed to participate. So if you have any questions during the webinar, which we hope there are lots of questions, we ask that you submit them through the Q&A feature on Zoom. They can be submitted anonymously, or you can submit with your name, whichever you prefer. We will address these questions toward the end of the program. And we look forward to a really lively discussion. However, if we run out of time and don't get a chance to address all the questions that are asked, we're more than happy to follow up with you directly after the webinar. Also, the webinar will be recorded, so a shareable link will be provided to all the viewers after the event. So with that, I want to get started with just a few introductory remarks. So here in the GI Cancer Center, and really at the Mass General Cancer Center in general, we really operate under a bedrock principle of bringing people together to improve care for patients. Now, this includes the way we deliver clinical care. We bring together people from different specialties, from surgery, radiation oncology, medical oncology, and so forth, so that we can deliver seamless multidisciplinary care for our patients. But also, we take this same approach when we take on important research questions. And our major philosophy is combining those leading clinicians and clinical investigators really on the frontier of therapies for our patients with some of the premier scientists and cutting edge scientific technologies to really bring this together into something that we call translational research. What translational research represents is really translating scientific discoveries into the clinic and really designing treatments based on rational scientific discovery. And this is something that we believe has really helped us accelerate innovations in cancer care. And while we do this really throughout the GI Cancer Center, what you're going to hear about today are some examples of this type of really groundbreaking interaction and some of the advances it is leading to from a really phenomenal lineup of speakers. So joining us today, we have Dr. Sam Klempner, who is a gastrointestinal medical oncologist, who really leads our esophagogastric efforts from medical oncology here in the GI Cancer Center. He works with a multidisciplinary team to optimize and individualize treatments across all stages of GI cancers. We're also joined by Dr. Ted Hong, who is the director of gastrointestinal radiation oncology at Mass General, and who has research interests including proton therapy, targeted therapies, and biomarker developments for GI cancers. And he's really been just a pivotal member of the GI Cancer Center who's made many significant breakthroughs to the use of proton and radiation therapy for several GI cancers. We're also lucky to have Dr. Chloe Villani, who is the director of the Single Cell Genomics Research Program in the Center for Cancer Research, as well as the Center for Immunology and Inflammatory Diseases here at Mass General. She works with a multidisciplinary team of immunologists, geneticists, computational biologists, and physicians to establish a comprehensive roadmap of the human immune system. And finally, we're lucky to have a special guest today, Dr. Jesse Boen, who is a principal investigator at the Broad Institute of MIT and Harvard Medical School, and the chief scientific officer of Breakthrough Cancer. And Dr. Boen collaborates with a team at Mass General to bring the power of genomics, specifically functional genomics, to bear on living samples from cancer patients with a particular emphasis on rare and underrepresented tumors. So this is going to be a very exciting program. And with that, I'd like to hand it over to Dr. Klempner.
All right. Thank you, Ryan, and thank you, everyone, for taking the time out of your day. Today, you're going to hear about some of our key team members in our program about how we're trying to tackle some of the key questions and unmet needs in gastric and esophageal cancers. I know several of you have been personally touched by this terrible disease, and I hope the message of hope and excitement comes through in the webinar. I'm going to try to set the stage and then turn it over to our other team leaders to expand on the approaches. So I was actually 24, and I remember sitting at a computer in medical school studying when my dad called to say my mom was diagnosed with cancer. My mom had actually been a nurse at MGH since before I was born, and now in a span of hours was becoming a patient at the cancer center. There was never any doubt about where she was going to get her care, and like many of you, I sought to try to educate myself about her disease. Uh, Thanks to the excellent care provided, she survived almost 2 years before passing comfortably. It's now 16 years later, and I'm still bothered by many of the fundamental questions that are facing us in gastric and esophageal cancer. And I carry her ID badge here as a reminder that we just need to do better. So when the average patient is diagnosed with gastric or esophageal cancer, their disease is either locally confined or already spread to distant organs. Patients with locally confined disease generally receive neoadjuvant therapies involving chemotherapy and or radiation therapy to the primary tumor prior to surgery. Really, the goal here is to eradicate and sterilize both the area where the tumor started in the esophagus or stomach, but also try to deal with small areas that we can't see with our current technologies, and these are called micrometastases. Unfortunately, our current approaches really don't adequately deal with these hidden areas, and over 50% of our patients will recur within 3 to 5 years after being treated with the goal of cure. So why does recurrence happen at such high rates in our patient population? And to give you some context, this is actually far inferior to what we see for common cancers like breast and prostate cancer, where we have more effective therapies. Led by Dr. Hong, we're actually investigating several novel approaches to improve our neoadjuvant therapies with ultimately the goal of improving cure rates. Some of the technology platforms we're using include detecting minute fragments of cancer DNA in the blood, as well as a deep dive into how the immune system may shape the response to our therapies. And you'll hear much more about this from Dr. Hong. Due to the very high recurrence rates and the fact that about half of our patients present with stage 4 disease, we have a big focus in building a framework to improve treatment options for advanced patients. If you've known anyone who developed fluid buildup in the abdomen related to cancer, then you clearly understand what an impact this can have on someone's quality of life and symptoms. It's about 40% of our patients with gastric and esophageal cancer will ultimately get fluid in the abdomen, and this really impacts the quality of life and is associated with a poor survival. This fluid, when it builds up, is called ascites. Normally, we all have a little tiny bit of fluid in the abdomen, and this serves like a lubricant. But these patients who develop ascites, they have a cancer-driven increase in this amount of fluid, often to the point of many liters, and really need to have this drained weekly, if not every couple weeks, just to achieve some level of comfort. Unfortunately, patients with ascites are very underrepresented in all of our clinical trials, and the limited data we do have suggests that our current therapies really don't work in the abdominal compartment, in the peritoneum, the lining of the abdomen, or in this fluid. So despite appreciating this clinical problem for some time, there remains near a complete scientific blind spot regarding the biology. It's actually quite remarkable how little has been done. There's thousands of research papers detailing the composition of tumors to a pretty high level, including specific mutation patterns and presence of immune cells, but there's literally only a handful of papers looking at this fluid. This problem actually affects about 400,000 patients a year with gastric and esophageal cancer. And so to tackle this effort, we've developed a dedicated effort to really try to understand this clinical problem. And led by Dr. Villani, we really have an intense effort to explore the components of ascites at the single cell resolution level, so a very detailed and granular understanding. Ultimately, this has the potential to really nominate targets that are specific to this population and try to understand the biology. And you'll certainly hear Dr. Villani expand upon the exciting approaches. So perhaps the largest problem is that outside of very rare situations, and despite years of cancer research, oncologists are actually quite poor at making predictions about how likely a patient is to respond to a given therapy. For the average patient coming through the doors of any cancer center, we can only make educated estimates based on prior clinical trials, which may not accurately represent each patient. So why are we so bad at what seems like such an important part of our job? So a big portion of the problem comes from how we study the cancers in the first place. Most of our research relies on cells that are made from patient tumors, and many of these cell lines have been grown on plastic for long periods of time. This poses a significant problem because what survives growing on plastic and gets frozen and thawed many times over may not be a good representation of what's actually happening in the patient. I think it's pretty clear and obvious to see why making treatment predictions from a model that doesn't reflect the real thing is highly problematic. I mean, this could be like driving a bunch of pickup trucks to decide which is the best sports car. They're really not good representations. Dr. Boen and the team at our collaborating site, the Broad Institute, are really some of the leaders in tackling the problems of cancer dependencies and models. And so together, we're exploring some of the advantages and exploiting some of the advantages of free-floating cells and ascites to build approaches to improve response prediction, ultimately maybe even at the patient level. And I have to say, this has been one of the most fun collaborations of my entire career so far. So personally, I've gone from a scared and often hopeless medical student sitting with my mom in the MGH Cancer Center to a GI oncologist here that's full of hope and gets to work with one of the best teams in the world. So I'll now let you learn a little bit more about each of the efforts, starting with Dr. Ted Hong.
Thank you, Sam, for that introduction and that really touching personal story. And also welcome to our folks who are listening to this. I know that there are some friends and patients of mine who are here, and I hope that you learned something and enjoy this presentation. What I'm going to talk a little bit about today is how do we get to more cures in the context of patients who have localized or locally advanced disease. As is true with many GI cancers, when we think about the way we treat patients with GI cancers, we think about surgery as a primary modality that gets us to a potential cure. But we know, and as some of you who have met with me know, that surgery is usually insufficient to have a really good shot at being cured of the cancer. And there are a lot of strategies that have been evaluated across GI cancers, looking at treatments afterwards, looking at treatments before, to try to decrease the risk of the cancer coming back either elsewhere in the body in the form of metastases or growing back where the cancer started, what we call a local recurrence. Some of the things that we've learned over the past couple decades is that the treatments are better tolerated when we do the treatments before the operation as opposed to after the operation. And not only that, in many situations, the cure rate is higher. And this has led to an approach that is commonly used in esophagogastric cancer, where we will deliver radiation and chemotherapy prior to the operation in order to try to improve the chances of a clean resection and subsequent cure. However, even with that standard approach, we know that over half the patients who undergo that standard approach will unfortunately develop their cancer either elsewhere in the body or coming back in the area of the operation. An approach that we've really led in a related cancer, in pancreatic cancer, to try to improve the cure rate there has been a strategy of total neoadjuvant therapy, where we give all of the chemotherapy first, followed by the radiation and chemotherapy, with an attempt to really accomplish two goals. One, to decrease the spread of the cancer, improve the cure rate that way. And two, increase the likelihood that we've gotten rid of the cancer completely where it started. And based on a couple studies that we did here at MGH, we have transformed our ability to get folks to a curative operation in pancreatic cancer. Now, as you know, I treat a lot of esophagogastric cancer with Dr. Klempner, and we thought that we should be evaluating a similar approach in esophagogastric cancer as well. And this led to a small pilot study that we did looking at a regimen called FOLFIRINOX, an aggressive 3-drug regimen, followed by radiation and chemotherapy before surgery. And we learned a few things from that. This was a very high-risk group of patients, many of whom had spread of their cancer to lymph nodes that are outside where a surgeon would typically think about operating. And in spite of that, we found that in about a third of the patients, we actually sterilized the cancer completely. And not only that, we have a number of patients who would have been deemed incurable at the time of presentation, who actually are now about 4 years out without their cancer ever coming back. And this got us thinking that this is an approach that we should be looking at in order to try to expand who we can cure with esophagogastric cancer, both in terms of getting them to an operation and not having their cancer come back, but also maybe if we could sterilize the cancer completely, we could even spare some people of this very large operation. So in collaboration with a pharmaceutical company who had designed what we call a nanoliposomal form of a chemotherapy agent called irinotecan, Dr. Klepner and I put together a chemo regimen and a strategy where we would look at a 3-drug regimen with FOLFOX plus this new drug, follow it with radiation and chemotherapy in the hopes that we would further improve our ability to cure patients. And we're really excited about this study. The study was recently IRB approved, and we expect that it will open within the next week or two. But based on this framework of this study, our group at Mass General also applied for a grant from the organization Stand Up to Cancer with the hopes of leveraging this prospective clinical trial to get some baseline information on, was it possible for us to do a better job detecting either early cancer or minimal residual cancer in the esophagogastric tract, as well as evaluate some of these blood markers that Dr. Klepner was referring to, where we can detect tiny fragments of cancer. So we had a better understanding, who did we eliminate the cancer completely, or in whom there is a little bit of cancer left. So based on this clinical trial, we now have a framework where we can begin to ask all of these really interesting scientific questions in collaboration with this outstanding team that we have at Mass General to try to untangle what can we do to further individualize care, make care more aggressive in some situations, or even back off on things like surgery in some cases to really get the right amount of treatment to each individual patient. We are now also moving this strategy in the hopes of curing more patients beyond locally advanced disease to early metastatic disease, where patients sometimes will have 1 or 2 spots that have spread to the liver or the lung, and aggressively go after them with either focused radiation or even surgery in an attempt to try to cure patients that we did not think previously curable. And again, leveraging these blood-based techniques that were really pioneered by Dr. Corcoran to understand, were we impacting, making a biological impact on the natural history of a patient's journey through their cancer. The one other thing that I would like to finish with is that we're always looking to try to do our treatments more safely. And more safely now, it turns out, also means protecting a patient's immune system, as immunotherapy has now entered the standard of care as being something that can improve the cure rate when given after preoperative therapy and surgery. But we know that our treatments can be very effective in a bad way at depleting the immune system. So we are looking at a technology called protons, which you know MGH has been a leader in for the past several decades, as a way to spare the immune system and keep the immune system functional. And there are many ongoing efforts that we have specific to esophagogastric cancer that we're working on to try to protect this very important immune niche. With that, I would like to say that our clinical investigations really provide a great framework, not to just help our patients today, but to help us learn what we need to be improving upon in the future. But we really need great, brilliant scientists to help us untangle exactly what is going on and understand the biological impacts of our therapies. And with that, I would like to turn it over to Dr. Villani to talk a little bit about what I mean by untangle the biologic consequences of our treatments.
Thank you, Dr. Hung, for the introduction. So it is my pleasure to tell you a little bit more about some of the cutting edge technologies we're leveraging to try to solve some of these big biomedical problems. Dr. Klepner introduced you to some of the challenges they are facing in the clinic, including that 40% of the patients with gastroesophageal cancers develop ascites, this accumulation of fluid in the abdomen. Yet, as Dr. Kleppner mentioned, we know very little about what is in the fluid and why these patients are doing so poorly. Are there cells in the fluid? Are there molecules in the fluid that makes this bath where the cells may be floating, what we call immunosuppressive? So that would be these molecules that your immune system is capable of secreting to suppress the attack against your cancer. Or are there molecules that makes this fluid very inflammatory? And so these questions are important to answer if we want to try to narrow down potential therapeutic targets. MGH, as many of you know, includes teams and leaderships that are visionaries. And they've invested in a series of cutting edge technologies that are now available to answer these questions and brought me over to start tackling some of these big problems. One of these series of technologies are called single cell genomics. And so I'm gonna use some analogies to try to explain the power of these amazing technologies. Let me share my screen. So we're going to use a fruit salad here as an analogy. And so where every single piece of fruit here would represent a specific cell, and could be a cell in the tissue. And today we're going to talk about cells in this fluid that builds up in the abdomen. And so traditionally, over the last few decades, the way we've been analyzing the composition of this fruit salad or cells in your tissue is by basically blending everything together and creating this fruit smoothie and basically measuring the average of the signal. That's problematic because not all the cells may be driving the disease. So if I take as an example these 3 blueberries here, if you blend them in your fruit smoothie, and these 3 blueberries would represent your pathogenic cells really driving the signal. If you blend them in a fruit smoothie and I ask you to taste my fruit smoothie, and I ask you how many blueberries are there? Can you tell me if they're from Maine or from California, from Canada? Are there some that are rotten? It would be impossible for you to answer this question. And there's the same problem when we mash all the cells together to try to identify these pathogenic cells. We now have these amazing technologies that are called single cell genomics, which allows us to identify every single piece of fruit in this case, or every single cell in a particular fluid or tissue. Not only can we identify them, we have computational tools to regroup them all together. So I can tell you how many pieces of apples, how many pieces of kiwis, I can compute my number of blueberries here. And so I can also quantify the number of cells I have for each of them with these powerful technologies. These are new technologies. The first time we managed to sequence a single cell was very recently in 2009, and that was like a single cell. Now, 2012, and now in 2021, we're now capable of analyzing the profile of hundreds of thousands of cells in a single sitting. So amazing progress in the recent years. Beyond telling you, for example, that I have apples in my fruit salads or specific groups of cells in my tissue, we can go a step further. So I'm gonna take apples as an example as a good Canadian kid who grew up on orchards. I know my apples quite well. And so apples here would represent a group of particular cells. So I'm taking an example of a group of immune cells that are known to have propensity to attack your tumor cells when they're in good shape. And so there's many different type of these cells, like there's many different type of apples, right? So I'm showing you some examples of apples here. And I don't know if you know your apples as much as I do, but there are some apples that are good for applesauce, right? Some others are good for cooking with meat or for juice. They each have their particular properties. It's the same when we do a deep dive in the particular group of immune cells. And not only can we do that, we can focus with our technologies on particular cell type, or in this case, particular apple, and tell you if it's a pathogenic cells or if it's a healthy, juicy apple, if you wish. And so we can create this very detailed picture of every single cell in a tissue and body fluid and tell you if it's a good cell or a bad cell. So here I'm showing you a juicy apple or one that's completely rotten. And so how do we do that? So we have these interesting technologies, and if some of you would like to hear more about it, I'm always happy to discuss this in further detail. But it's the principle of emulsion. It's like when you try to do salad dressing at home, and you layer oil and vinegar, and then you shake it to make these little droplets. So we use the same principle. Obviously, we're not shaking our cells with oil, but we are using machine that can create these drops. So what you're seeing here, here you cannot really see it, but there's oil passing through this liquid, which is what creates these droplets. If you focus on here at the bottom, you're going to see a very small clear dot go by. That's a cell that we pass through our microfluidics. And then the very big dot is a gel bead that can squish. And here you have a snapshot of what is in the drop. So we're capable of capturing a cell in a drop. You can imagine we can do thousands of them. And we capture them with this bead that contains reagents that once we heat it up, it melts the beads and can open up the cell. It can lyse the cells so that we can then capture the genomic material. And that's because in the fluid that goes through, we have the reagents that will allow to amplify the genomic material. And that's the beauty of these technologies. We have now, working closely with Dr. Kleppner, established a framework in the clinic with a very talented fellow, oncology fellow that him and I share, Dr. Boen, who has been collecting ascites. Working closely with Dr. Kleppner, we've now analyzed over 6 patients with these technologies, looking at the content of the ascites, and have discovered that patients are heterogeneous. And then some patients have a lot of immune cells in their ascites, and some have a lot of cancer cells. So we have also optimized and developed our experimental and computational frameworks to achieve this. We're at an exciting time. We're ready to scale up our effort to analyze a greater number of patients. And that is also why we're currently fundraising for these efforts. It's minimal risk because we've already established all of our frameworks. And that will allow us to nominate new therapeutic targets, targeting both cancer cells as well as stimulating immune cells to attack the cancers. And as we nominate these therapeutic targets, we'll need to move on to validate them with platforms that appropriately represents the different type of cells that are in ascites. And that will be done working closely in collaboration with Dr. Boen at the Broad Institute. And so I'll let Dr. Boen tell you more about this now.
Thank you, Chloe. I really appreciate that. It's wonderful to be here, and I'm really excited to see so many folks joining the webinar this morning. So maybe just to pull the frame up a little bit from these amazing technologies and clinical situations that you've heard about, my reaction is that this is an unprecedented moment for science in the United States. If we think about what we've all been through and what we've witnessed being achieved on a national level to fight the coronavirus just in the past 14 months, I think it's been clear the potential of working together, making investments in new technologies and new frontiers, and working collaboratively across conventional barriers between laboratories, between organizations, to achieve things that we thought previously were just totally impossible. Making a coronavirus vaccine in 12 months. I think most infectious disease researchers would have said totally impossible, but a series of activities and an important moment of urgency helped to usher that into the state of reality. And I think in the face of cancer, that same urgency, of course, is felt every single day and every single week by patients that all of us interact with. And I think at this moment, we have to emerge out of the coronavirus pandemic with a renewed sense of urgency of working together across the entire ecosystem to bring cures to patients as rapidly as possible. And that's one of the reasons I've been most proud of working very closely with the MGH team, because it's that same sense of urgency and it's that same sense of collaboration that they've always brought to these types of projects. So what we're trying to do is work collaboratively to address what I think is one of the most fundamental challenges in cancer, which is how do we, over the next decade, provide an oncologist with an information resource that enables him or her to read off of the genetic profile of the patient's tumor in front of him or her and make a prediction, an accurate prediction about which drugs or which combination of drugs to prescribe for that patient. It's a very simple concept, which, as many of you know, is the foundation of what's called cancer precision medicine. But it's a concept that to date just simply doesn't work for enough patients with gastroesophageal tumors, probably only 10 or 20%. And whether we're talking about a chemotherapy, a standard, cytotoxic chemotherapy, or a targeted therapy, or even one of these new immunotherapies that's working quite well, I think all of us are quite confident that the reason that certain therapies work for certain patients is encoded in the biology of that patient's tumor. And it's our jobs to figure out the connections between the molecular profile of the tumor and that response profile. Now, the standard way of doing this, of course, is to have a scientist create a hypothesis and test that hypothesis in a clinical trial. And that tried and true method is completely appropriate. It's just tremendously slow. So working closely with Dr. Klemner and the rest of the team here, we had a different idea. And we said, what if we could use living pieces of tumors, of gastroesophageal tumors, just a few hours from the patient, and grow them in the laboratory and use those living bits of tumors to be part of an activity to test every possible drug and every possible way of killing cancers in the laboratory, and produce a lookup table. We call it a dependency map, but kind of a Google Maps of all possible ways of killing all possible gastroesophageal tumors. Could living bits of gastroesophageal cancers from MGH come across the street, across the Charles River to the Broad, where we have a laboratory to grow out these patient cultures? We call them organoids. They're kind of three-dimensional cultures that grow in dishes. Could we do this not just with one patient, not just with 10 patients, but ultimately with hundreds of patients, and produce a map of the appropriate scale and resolution so that a clinician of 2030 would have this information resource in his or her pocket? So I'm pleased to say that that work is going extremely well. We've done this with over 100 patients to date. The success rate of actually growing these three-dimensional organoid cultures in the lab is actually quite a bit higher than historically was ever possible. It's one of the great paradoxes of laboratory science, I think, that cancers grow like gangbusters in patients' bodies, but when scientists have tried to grow them in the lab historically, only about 1%, or sometimes even less than 1%, of those cancers even grow in dishes in the way that Dr. Klemner mentioned before. But now, using these new organoid technologies, where cells are plated in a different way with a different cocktail of factors, upwards of 40%, sometimes even 60% of these gastroesophageal cancers can grow in the laboratory. And that is allowing us to capture a much more representative swath of patients, both in terms of their genomics, in terms of their different clinical presentations, in terms of their ethnic diversity and backgrounds. We need to make sure that every patient with a gastroesophageal tumor is somehow captured in this map. Once the organoids and the cells grow in the lab, there's a series of platform technologies that we've set up to put these cultures through their paces. We've created a drug repurposing library of over 6,000 drugs. This about 12,000 drugs have ever been tested in humans for any disease. And the team has collected about half of these and has automated these in a robotic platform that enables them to be tested against new cancer cultures. And so we routinely test about 6,000 drugs on new cells growing in the lab to see if any existing drug, perhaps developed for another type of cancer, could be repositioned or repurposed for a particular subtype of gastroesophageal tumors. But only about 5% of all of the proteins, all of the pieces of machinery in a cell are even targeted by any drug that any company has ever produced today. So that's where another technology, you may have heard about the Crispr technology, has become so powerful. So Crispr, as you may know, is an incredibly sharp pair of molecular scissors that allows scientists in the lab to snip out each gene one by one using a series of technologies that were originally engineered from the way bacteria defend themselves from viruses. And CRISPR allows us to cut every gene one by one and then see what happens to the cells in the lab. Do they die or do they stay alive? And so the way we build this dependency map is with patient cultures, with many, many drugs, and with CRISPR to cut every gene and look for new targets for which there aren't yet existing drugs. And so this work is moving along quite well. We've already identified a whole host of new targets, and we're eager to begin validating these in patient samples. The next question becomes, how do we validate these prior to enrolling patients in a clinical trial? We want to be sure that the best predictions are being made and they're not just fake outs of cells growing in dishes in the laboratory. So working closely with Dr. Klempner, we've just begun, just in the first 18 months, using the power of single cell genomics that Dr. Villani just mentioned, and the power of high content cellular imaging. Imagine a powerful microscope that is taking a picture of every cell in a dish, and not just saying it's a cell, but looking at thousands of morphologic features on every cell. Is it an immune cell? Is it a cancer cell? Is it dying when we test it with a drug? Is it staying alive? So as these new gastroesophageal clinical samples are coming across the river from MGH into the lab, we're testing drugs and we're building a platform that we call the biosensor platform to try to see whether we can reproducibly test a drug hypothesis or a CRISPR hypothesis that came from the dependency map, came from these long-term cultures of these tumors, and test this hypothesis in a faithful model of a gastroesophageal patient's tumor, maybe within the first 72 hours. This is a concept that scientists have been thinking about for 30 or 40 years, but Dr. Klempner and I feel that perhaps the time simply wasn't right. We didn't have enough genomic predictions. We didn't have the amazing collaborations across the Boston area, and we didn't have the amazing revolution in single cell genomics that Dr. Villani mentioned. And by working together across institutions, we're trying to build this biosensor platform to test predictions. We, at first, hope that this platform will be useful for invalidating or validating some of the drug target hypotheses that are emerging from the dependency map. And these are the ones that drug companies should build drugs against, and we should advance into clinical trials. But we also hope, although it's a fantasy at the moment, but we hope to usher in a future 5 or 6 years from now in which these types of biosensor tools could be used to validate and confirm molecularly guided predictions for individual patients with gastroesophageal tumors. But the work ahead of us requires a lot of work on reproducibility and confirmatory studies to confirm that even these rapid tests ex vivo are actually predictive of patients. So I'll stop there, but I think we're extremely excited to look at the decade ahead and harness the power of living gastroesophageal tumor samples and have these be an important part of addressing the precision medicine challenge for all patients with this horrible disease. And I'm just so happy and proud to be part of this great MGH team. So I'll turn it back over to Ryan, I believe, to lead us through some Q&A.
So a big thank you to all of our panelists for these outstanding and incredibly interesting talks. So I'm already getting a bunch of questions pouring in, and we really want to dedicate the rest of the time to question and answer from the audience. So just as a reminder, please do submit any questions you have through the Q&A function. It'll give you the option of using your name or submitting it anonymously if you're more comfortable doing that. So with that, I want to start with the first question. And this I'll direct to Dr. Klempner. How do you go about designing potential treatment combinations? Is it primarily clinically driven from patterns in patient cases and data? And maybe I'll add to that saying, how much of the time is it derived from some of these laboratory studies that we heard about today?
Yeah, this is a great question. I mean, I think there's how we currently do it and how we would like to do it are two very different things. But yes, currently a lot of it is driven by evidence. Evidence from clinical cases and clinical trials is really what shapes how we currently care for patients. But what we'd really like to do is increasingly integrate more information about an individual patient's tumor. And so routinely, we will get a sample of tumor and we will look for sort of common targets. And that will complement a standard therapy. We might add a targeted therapy or an immune therapy based on some observations. But as you've heard, those are quite crude predictions. And so we do add them, but the majority of the time, we're not clear if it's adding anything to the therapy for an individual patient. It's very hard to tease that apart. So the answer to the question is yes, we use primarily clinically driven data currently, but as we go forward with this team, really the goal is to integrate more and more molecular data so that we can make better combinations and better predictions for every individual patient. I hope that answers your question.
Great. Thanks, Dr. Klempner. So the second question, I think I'll perhaps maybe direct toward Dr. Hung, but obviously if any panelists want to jump in and offer some additional thoughts. The question is, what is being done to improve detection limits when there are rising CEA measurements? And just to clarify that for the audience, CEA is a common blood-based tumor marker, a protein tumor marker that we often use to track many GI cancers. And is there a need to identify where the cancer is in the setting of these rising CEA? So Dr. Hung, maybe you want to take that question and talk a little bit about some of the liquid biopsy approaches that you had discussed in your talk as well.
Thank you for that question. That's a really important question, and I'm sure that some of you on this call have dealt with the roller coaster ride of what happens when we use these tumor markers like CEA to give us a sense of, are things quiet or are things potentially becoming active again? Now, the problem with CEA primarily is that there's a substantial lack of what I would call specificity, meaning there's a lot of different reasons why the CEA can go up. And as an anecdote, I have a few patients who have had with various kinds of cancers who time has told us that they're cured 10 years later, but they went through this scare where their CEA went up dramatically. And then we get a bunch of scans, we get PET scans, we don't see anything, the CEA is still high, and all of a sudden for no good reason, the CEA drops again. And I think what that really speaks to is the limitations of what we can learn from CEA. CEA is really circumstantial evidence that we take in the context of other data like how's the patient feeling and what do the scans look like. And the scans do remain the gold standard. But to Dr. Corcoran's point, that is exactly the reason why our group is investing so much effort into some of these new liquid biopsy technologies, particularly circulating tumor DNA, where that has the benefit of being highly associated with the cancer. So we would know if the fragment, that DNA that we see in the blood, we would know if that was related to a cancer versus not to a cancer. And that could potentially give us greater insight into, is there active cancer or not? So, not to get too deep in the weeds, but there's a lot before we decide that we're going to act on a positive ctDNA test. There's a number of steps that a number of investigators, and this is very much a high priority in our group, in terms of validating, does an intervention just at the time of a positive ctDNA test impact how a patient is going to do in the absence of something on scans? It seems like an obvious question that if the ctDNA is high and we throw some therapy at it, that we're going to be able to cure more patients. But you can imagine that what it really gets down to is, are we giving the right therapy? And are we pushing that ctDNA level down to a point where it's a cure as opposed to just getting it to below a threshold where we delayed the cancer coming back again? And these are exactly the type of studies that we're doing in the context of our clinical trials across a variety of different GI cancers, that we are trying to determine if we can replace the scans with this blood test that is more accurate than a tumor marker, and will early intervention lead to more cures? But that is exactly the thrust of what this multidisciplinary group is trying to accomplish right now.
Great, thank you, Dr. Hong. And maybe I'll just pivot for a little follow-up to Dr. Klempner. So, a lot of the things I think we talk about in this webinar, there's a lot of very forward-looking, ideas that may sometimes seem several years off, obviously important ideas. But just to make a point, some of these ideas we're actually bringing into clinical use today, and really, I think this group has been on the forefront of integrating circulating tumor DNA and other liquid biopsy technologies. And so maybe Dr. Klemper, do you want to actually perhaps just briefly touch on how we're actually using these technologies in the patients that we see every day in clinic?
Yeah, sure. Definitely. Any of you who have passed through the clinic have probably been asked to or discussed with one of your providers the role for circulating tumor DNA. So this is, again, fragments that are derived from cancer cells that we can use to, one, detect mutations that we may have drugs to target against. So we have several ongoing clinical trials where if a patient presents with a new cancer, we would offer them a blood-based test and we may find a target which might make them eligible for a given clinical trial. So that's one way that we're integrating this clinically currently. And then when patients begin on a therapy, we're using that same information to see, is the drug modulating the target? So for example, in gastric and esophageal cancers, HER2 and EGFR and MET are some of the targets. These are just names for genes that we see in subsets of patients. So if a patient comes in, they get a blood test, and then 4 to 6 weeks after starting therapy, we may consider repeating that same blood test, and we can actually measure decreases in the amount of that specific mutation in the blood for a specific patient. So we can give, as Dr. Hong said, a more sensitive and specific answer to, are we killing cancer cells with our combination? Perhaps even earlier than we would get an answer from a scan, which is often done a couple months into therapy. So we routinely integrate this into patients to, one, identify candidate targets to go after that we're already using clinically, two, to monitor the efficacy of our therapies. And then one of the areas we're exploring most interesting is similar to what Dr. Hong said we're already using, is that after you finish therapy and we think you're done, is there anything left over that we can detect? And that's something called minimal residual disease. And we have a large effort to collect blood to study how does this impact our patients and what can we do about it. So I think there's already several routine ways we're using these technologies, but a lot of enthusiasm about where we can go next with the same platforms.
Thanks so much. And I think it's just remarkable to think that just from a simple blood draw, the same tube or two of blood that you might have drawn when you get routine labs, that all of this can be done. It's a pretty incredible thing. So we have another question coming through. The question starts with a statement. The work with Dr. Villani is fascinating. And I have to say, I 100% agree with that statement. And the second part of the question is, effective immunotherapy treatments are sorely needed in this space. Also a statement I totally agree with. About how long do you think it will be in years before we start seeing viable options for this population?
I imagine that question is dedicated to me.
I'm
going to take
absolutely
a stab
for
at
you,
it.
although I think
but I'll invite you and Dr. Kleppner and Hong, of course, to join as well. As things stand, I think realistically over the next decade, we can go from target identification to clinical trials, but there's multiple ways of accelerating the process. Dr. Boen gave you the example of COVID-19 research, how within a single year, the scientific community came together, got access to a large amount of funding, and managed to generate a vaccine that all of us are now eligible to receive. And so similarly, because we have all the framework to analyze these Ascites samples, with more funds, we could analyze these patient samples that we already have much more rapidly and get to target identification within a 1 to 2 year span because we have access to these samples. Another way we may be able to accelerate the process is to drug repurposing. There's already a lot of immunotherapy drugs out there. We don't necessarily need to engineer new ones, not only in the realm of cancer, but also in the realm of rheumatology research. And so drug repurposing based on our target identification could help speed up the process towards clinical trials. And I'll of course invite my peers to add to these comments.
I would maybe add, it's a clinical problem where we don't really have any standard therapies. There is no approved therapy for ascites from any cancer. And this, we're talking about gastric and esophageal cancers, but this has the potential to extend to other cancers because fluid in the abdomen occurs in multiple cancers, ovarian cancers, colon cancers, pancreatic cancers, etc. And so when there isn't an existing standard, unfortunately, the bar is very low. So I think there's the potential to find targets that would be identified from Dr. Villani's work on the immune side and as well as on the cancer side, and then maybe even rapidly test them with some of the work that Dr. Boen is doing and bring that back to a patient. You know, that's sort of the not really reverse translation, but the true translational science where we have a patient sample, go to our colleagues and come back to patient with some candidate targets. I would agree with Dr. Villani that with the ability to scale up these efforts, we can define sort of a landscape of targets that we could go after in probably a relatively short period of time.
And if I may add, unlike other research institutes where scientists like myself live in an ivory tower, here you have an integrated team where scientists like myself and Dr. Boen work directly with clinicians and oncologists that are in the front line. So our research is very much grounded to the clinical problem being faced. So we are trying to answer these exact questions and be very pragmatic. And beyond that, Dr. Klempner has some very creative idea about directly injecting potential drugs in the peritoneal cavity. So we have these questions constantly in the back of our mind.
Yeah, I think that's a great answer. And I think, a really important point about these types of collaborations, which really, I would say really don't happen as much as they should, elsewhere, is really a key. And I think something that we think is key to really accelerating advances for patients. And I thought it was interesting that, the question was phrased, how long, parentheses, in years, do you think it'll take? Well, we're actually seeing this incredible single cell genomics revolution impacting our treatment, options and ideas now. I mean, we were actually integrating analysis with single cell genomics into many of our clinical trials, and that's informing clinical trials that we might open in a few months from now, or by the end of the year, early next year. So actually, in a time frame in some cases shorter than a year. So we really do believe that this type of integration is important to make these discoveries move more rapidly, since they're as we discussed, there's a huge unmet need for this. So one additional question coming in, again, that starts with a statement that I wholly agree with. It is nice to see Dr. Boen on this panel representing the Broad. I agree. Can you share a little more about how these collaborations form? I imagine it is a competitive space with intellectual property. How are these partnerships structured from that vantage point, and also funding? And I think this just goes to the issue we were just talking about, that these collaborations just seem so logical and to us just seem like a necessary thing. But traditionally research is very siloed, and this doesn't happen at other places. And I think breaking down those silos and getting people to work together is important. So maybe Dr. Boen, do you want to take the question? How do we approach these incredible collaborative teams like this?
Yeah, I mean, maybe to start with philosophy, I think, we had a fantasy about cancer research in the 1980s or the 1970s that solving cancer was the sort of thing that, a very smart man or woman and people in their lab could do on their own without engaging others, without working collaboratively. And that fantasy, I think, was really shattered, especially after we discovered the complexity of cancer emerging from the human genome era, after we realized that, cancer is like 1,000 different subdiseases with many components. I think all of us here have grown up as part of a new generation of cancer researchers that recognize that the really important challenges require activities that are at the intersection of different disciplines. Often, as Dr. Corcoran mentioned, the intersection of the clinical world and the laboratory world, or one technology and one area of chemistry, and often between institutions as well. And so I think the concept of the Broad is very similar to the concept that Dr. Corcoran laid out for MGH, which is how do we free up scientists to follow the science and articulate big challenges and solve those challenges regardless of where their collaborators might happen to live. And all the Broad it helps researchers do that as much as possible. In terms of the specific questions about IP, I mean, it's the same question within an organization like MGH. I think there's a master collaboration agreement which describes how these decisions get made, and it basically follows that the person that the team that created the intellectual property files on it. In terms of funding, the different projects follow different paths. This particular project is a blend of philanthropy, and that philanthropy always begins these projects, and then government funding comes in 10 years later or something like that. And it's helpful to have multiple collaborators working together because those folks can often write different types of grants. But I think that the most fun science happens as part of teams. But just again to emphasize that the great innovations that I've observed over my career are always the result of a few people who are approaching the problem from different disciplines together with the liquidity of early stage philanthropy that actually sometimes provides that kind of tap on the shoulder to say, hey, go pursue that really interesting creative thing. Maybe it'll fail, but maybe it'll succeed. And writing a grant is a 10% shot at maybe getting funds after 2 years. And so that really slows down what really should be a free-flowing and iterative process. And so the specific answer to the question of how do these collaborations get started, it's almost always with the liquidity of philanthropy that helps them get moving, and then government or other types of funding follows on 6 or 7 years later.
So thank you. So I unfortunately, this has been an outstanding discussion, but we are running out of time here. And I do see there's a few other questions that we are going to be unable to get to. So I do encourage folks, if you, as I said at the beginning, if you have questions that you want to ask, please do feel free to reach out to us. But with that said, I want to thank you on behalf of the GI Cancer Center program and the Mass General Cancer Center to thank you all for attending this webinar today. I think what we've heard are just some outstanding, just innovative and collaborative efforts that we really hope and really expect are going to have a phenomenal and incredible impact on the way we treat gastric and esophageal patients in the very near term. And echoing something that Dr. Boen just said, I think much of the research that you've heard about today really has been spurred by philanthropy. Sometimes from some cases from generous gifts from people on this webinar. And I think that typically some of the most transformative breakthroughs that we see in science don't come from kind of safe classical research. That's the type that typically is funded by the NIH or the government. That it's often these kind of groundbreaking ideas that seem a little too risky, but are the ones that really pan out and move the field forward. And that's really something that philanthropy and support has been integral to pushing forward. And so, we really hope you've enjoyed this presentation, and we really hope that you would consider partnering with us on this important mission to solve this critical problem of gastric and esophageal cancer. And encourage you to reach out to us and to Megan Coons in the development office if you have any questions. And I really appreciate everyone. I want to thank everyone again for their time today and hope you enjoyed the session. And thank you again to our panelists for joining. Take care.